Thanks for joining us for this symposium. I'm Bill Rapisi, President and CEO of the Lymphatic Education Research Network. LE&RN's mission is to fight lymphatic diseases through education, research, and advocacy. In order to win a fight, you first have to join it. So we ask, please become a supporting member of LE&RN at lymphaticnetwork.org. And we hope you enjoy today's symposium.
Welcome to the 2022 LE&RN Symposium series. My name is Ying Yang. I am an assistant professor at University of South Florida in the Department of Molecular Pharmacology and Physiology. I would like to thank LE&RN for inviting me to present the research from my laboratory. I would also like to take this unique opportunity to say hi to all lymphedema patients and say thank you to all the scientists and the physicians who are working hard to find a cure for lymphedema. Today, I'm going to present our recent work on the function of a protein called Foxo3 in the formation of lymphatic valves, which has provided therapeutic implications for treating lymphedema. If you have any questions, please type them in the Q&A box. I will answer these questions after my talk. I would like to thank all the sponsors for the 2022 LE&RN Symposium series. This is the disclaimer from LE&RN. The title of my talk is A Novel Mechanism to Enhance Lymphatic Valve Formation, Therapeutic Implications for Lymphedema. Here, I'm showing you a lymphatic vessel with a fully formed valve, fully formed lymphatic valve in the lumen, which is the V-shaped structure in the middle. The valves have two leaflets, which makes the V-shape. These valves are crucial for keeping fluid in the lymphatic vessel to flow in one direction. When the valves are open, it allows fluids to pass, and when they are closed, the fluid cannot go back. I will talk a lot more about these valves later in my presentation. Before I present our data, I would like to give you an overview of the lymphatic vasculature, because I think this will help you to understand our experimental results later in my talk. The lymphatic vasculature is the major transportation pathway of tissue fluids. It absorbs and transports tissue fluids through lymphatic vessels and the lymph nodes. Then it collects all the fluids in the biggest vessel, lymphatic vessel in the body, which is the thoracic duct. The fluids will eventually be returned into the bloodstream through the connections between the thoracic duct and the subclavian vein. There are two types of lymphatic vessels are shown in this close-up view of the lymphatic vasculature. The lymphatic capillaries are responsible for uptaking fluids because these vessels are highly permeable, which allows fluids, macromolecules, and cells go inside of the lymphatic vasculature to form lymph. The downstream collecting lymphatic vessels are the major vessels that transport lymph. So one of the unique features of these collecting vessels is that they have smooth muscle cells wrapped around them. These smooth muscle cells can contract and drive the pumping of these vessels, which helps to move lymph going forward. Another feature of the collecting lymphatic vessels is that they develop valves in the lumen. As I mentioned earlier, these bicuspid valves act as the gatekeeper to maintain lymph only go in one direction, which is towards the lymph node in this image. Therefore, the lymphatic vasculature absorbs interstitial fluid and transports it to the bloodstream to maintain fluid homeostasis. The lymphatic vasculature is also important for transporting immune cells for immune response. Additionally, the lymphatic vessels in the gut villi absorb and transport dietary fats, which is crucial for lipid absorption. Since the lymphatic vasculature is the major transportation pathway of tissue fluids, healthy lymph flow is critical for the lymphatics to function properly. So what is driving the forward lymph flow? There are two mechanisms. One is the pumping of the collecting lymphatic vessels, which is driven by smooth muscle cells contraction shown in the top video. Through each pumping, lymph is moved forward. The second mechanism to keep forward the lymph flow is valves, shown in the bottom video. The valve leaflets open and close in response to the upstream and the downstream pressure of valves to ensure that lymph can pass but cannot go back. So when the valves are defective, it can cause leaky valves, which means the valve leaflets are not completely sealed tight. In another word, lymph can go backwards through leaky valve leaflets, which will lead to stagnant lymph. Most scientists and physicians agree that lymphedema starts with stagnant lymph, and a stagnant lymph triggers inflammatory response, which causes fibrosis and adipose tissue deposition. Everyone in this audience is probably familiar with lymphedema. Briefly, lymphedema is the accumulation of fluid in the tissue caused by impaired functioning of the lymphatic vasculature. Congenital lymphedema is caused by inherited mutations in genes that regulate lymphatic development. This is an image from a patient with lymphedema distichiasis, which is caused by mutations in Foxo3 gene. And Foxo3 is critical for the formation of lymphatic valves. Lymphocytography was performed to visualize the function of the lymphatic vasculature. As you can see here, dye was drained by the lymphatic vessels to the lymph nodes in the healthy patient, while a large amount of dermal backflow was seen in the lymphedema distichiasis patient. The presence of retrograde lymph flow indicate that lymphatic valves are dysfunctional in this patient. In addition to Foxo3, many other gene mutations that cause congenital lymphedema have valve defects in their corresponding knockout mouse models, as shown in this list, which indicate that valve defect is a causative factor for congenital lymphedema. Secondary lymphedema is also called acquired lymphedema, which is caused by injury to the lymphatic vasculature after birth. The most common cases of secondary lymphedema is from lymph node removal surgery during cancer treatment. Breast cancer patients are the largest population in developed countries that have secondary lymphedema. And similar to the congenital lymphedema patients, secondary lymphedema patients also showed dermal backflow in lymphocytography, which indicate that these patients also have dysfunctional valves. Since valve defects are strongly associated with lymphedema, my lab is working to understand how valves form and how to enhance valve function to improve lymph flow. How do lymphatic valves form? The current understanding is that valve formation is a stepwise process. It starts in the embryos. When the embryos develop between embryonic day 15 to 16, a cluster of cells in the lymphatic vessel wall begin to have increased expression level of 3 genes. They are PROX1, FOXY2, and GATA2. This step is called initiation. These cells soon will form a ring-like structure to establish the valve territory during condensation step. Valve leaflets form between embryonic day 17 and 18 with deposition of extracellular, deposit of extracellular matrix, which is called elongation step. Maturation step is when two valve leaflets finish forming. Each valve leaflet is composed of 2 layers of lymphatic endothelial cells with an extracellular matrix core in the middle. As I mentioned earlier, functional lymphatic valves maintain forward lymph flow. Vice versa, lymph flow is crucial for the formation of lymphatic valves. This study has shown that in the absence of lymph, the expression of PROX1, FOXY2, and GATA2 is not upregulated. Therefore, no valves form. Lymph flow is not only important for the formation of lymphatic valves, but also is required for the lifelong maintenance of the valve leaflets. The expression of FOXY2 is maintained by oscillatory lymph flow, and the loss of a FOXY2 gene results in valve regression, meaning valves disintegrate after they are already formed. These data indicate the pathological changes in lymphedema distichiasis patient that were caused by FOXY2 loss of mutations. So fluid shear stress from lymph on the surface of lymphatic endothelial cells is critical for regulating signaling transduction pathways inside of the cells, in turn to activate the expression of valve-forming genes, FOXY2, GATA2, and PROX1. This is an image from blood endothelial cells. In the blood endothelial cells, the mechanosensory complex on the cell surface mediates the endothelial cell response to fluid shear stress. So one of the downstream effectors of the shear stress signaling pathway is AKT, which controls cell proliferation, cell metabolism, cell migration, etc., through their protein kinase activity. Results from our previous study, as well as from many other labs, show that AKT activity is upregulated in the cultured human dermal lymphatic endothelial cells. When these cells were treated with oscillatory flow, indicated by the levels of phosphorylated form of AKT. So our question is that, what is the downstream target of AKT that controls lymphatic valve growth? One of the proteins drew our attention when we reviewed the literature. In many cell types, including the blood endothelial cells, AKT regulates the activity of a transcription factor called FOXO1. As a transcription factor, FOXO1 protein locates in the nucleus and binds to the DNA of its targets. When AKT is phosphorylated, phospho-AKT phosphorylates the FOXO1 protein. After FOXO1 is phosphorylated, it no longer stays in the nucleus. It leaves the nucleus and enters in cytoplasm to be degraded. In other literature, FOXO1 is known to repress blood endothelial cell proliferation. However, its role in the lymphatic vasculature was not known. So we decided to delete FOXO1 from lymphatic endothelial cells by crossing FOXO1 flophlox mice with PROX1 CREAR-T2 mouse model that was generated by Dr. Tai and McKinney, which is widely used to delete genes in lymphatic endothelial cells. In other words, we can delete FOXO1 in lymphatic endothelial cells by injecting a chemical at any time. Deletion can be induced in the embryos, in the postnatal pups, or in the adults. I will refer this mouse model as the FOXO1 lymphatic knockout in the rest of my talk. And meanwhile, we included a PROX1 GFP reporter mouse model generated by Dr. Yang Huang to visualize lymphatic vessels because PROX1 is expressed in all lymphatic endothelial cells. The entire lymphatic vasculature is in green fluorescent color. We first deleted FOXO1 in the embryos when lymphatic valves start to form and analyze the mesenteric lymphatic vessels, which is a lymphatic valve enriched tissue, when valves finished forming. And the first thing we noticed was that the lymphatic vessels were dilated in the FOXO1 lymphatic knockout, which is consistent with previous studies that FOXO1 represses cell proliferation. Therefore, cells proliferate more when FOXO1 is deleted. Then you probably already noticed the FOXO1 lymphatic knockout mesentery has many more white arrows pointed valve structures. Our quantification result on the right-hand side showed that deletion of FOXO1 significantly increased the number of valves per millimeter compared to the control mesentery. These data led us to hypothesize that FOXO1 could be a repressor for valve formation. We next used the whole mount immunostaining to analyze the expression pattern of FOXO1 in the mesenteric lymphatic vessels at embryonic day 16.5, when lymphatic endothelial cells become specified for valve formation by upregulating PROX1. When lymphatic, we stained the lymphatic vessels with PROX1 in green and the FOXO1 in red. We were excited to see that green cells are only in the valve specification sites, that is in the white squares. These cells are green because they lack FOXO1 in the nucleus, indicating that FOXO1 lost its nuclear activity in the specified valve cells, which supported our hypothesis that FOXO1 is a repressor during valve development. To gain better resolution of the valve structure in the bigger sized vessels, we next deleted FOXO1 in the postnatal pups using the same mouse model right after they were born, and analyzed the morphology of the lymphatic vessels 1, 2, 3, and 4 weeks after deletion. We were happy to observe many valve-forming areas at all 4 stages. Here, I'm using 1 week after deletion as an example. You can see 2 valve initiation areas in the Foxo1 lymphatic knockout mesentery, which are rarely seen in the control mesentery. These areas match the morphology of the valves that are under formation, as I mentioned earlier. To confirm that, we induce a new valve formation in the Foxo1 knockout postnatally. We quantify the total number of valves in the mesentery. The Foxo1 lymphatic knockout mesentery had significantly more valves than the controls. Then in this slide, I included the results from both 2 and 3 weeks deletion in addition to the 1 week deletion. Different from the embryonic deletion, postnatal deletion of Foxo1 did not increase vessel size, as you can see here, which indicates that the function of Foxo1 in vessel size control is developmental stage dependent. More importantly, consistent with the embryonic deletion data, the valve number per millimeter was significantly increased in the Foxo1 lymphatic knockout compared to the controls at all the stages. The postnatal results confirmed that Foxo1 represses lymphatic valve formation. By removing Foxo1, new valves grow. In addition to the mesenteric lymphatic vessels, we also observed increased number of valves in axillary lymphatic vessels. These data show that deletion of Foxo1 stimulates valve growth in more than one tissue type. In addition to deleting Foxo1 in the embryos and the postnatal pups, we also induced the deletion in the adult mice and tested whether Foxo1 deletion can induce new valves to grow when valves are not actively forming. Our results showed when deletion was induced in 7-week-old mice, the number of valves was significantly increased in 2 weeks. This indicates that the function of Foxo1 in lymphatic valve formation is not developmental stage dependent. So besides Foxo1, Foxo family member Foxo3 has been shown to have functions in the blood endothelial cells. So we were curious about whether deletion of Foxo3 would have the same phenotype as Foxo1 deletion. We used the same CROX1 PRT2 mouse model to delete Foxo3 and analyzed the lymphatic vasculature 2 weeks after deletion. As you can see here, deletion of Foxo3 did not induce new lymphatic valves to grow, and we did not see any changes in the number of valves between the Foxo3 lymphatic knockout and the control. We also generated a double knockout of Foxo1 and Foxo3 because that will answer the question whether the new valve formation phenotype from Foxo1 deletion requires Foxo3 activity or not. In a double knockout mesentery, we still observe many valve forming areas indicated by white arrows here, and the number of valves were increased in the double knockout compared to the controls. And this valve number was similar to Foxo1 single knockout. Therefore, with all the data so far, we have shown that Foxo1 deletion in the lymphatic endothelial cells can induce new valves to form, and this activity is independently of Foxo3. To further characterize the new valves that were induced by Foxo1 deletion, we performed the whole mount immunostaining and the confocal microscopy to investigate whether these newly formed valves expressed valve-forming genes such as PROX1, FOXY2, GATA2, KL4, and integrin for alpha-9. As you can see in the images, the newly formed valves express all these valve-forming genes, which indicates that these valves were in the process of normal development. There was also no difference in the process of smooth muscle cell recruitment. Moreover, these valves did not show any defects in the junctional protein expression. And the junctional proteins are important for maintaining the integrity of the vessels and the valves. These results indicate that deletion of Foxo1 did not induce defective valves. All the new valves appear to have normal expression of valve genes. Now you might be as curious as we were about how Foxo1 deletion led to new valves to form. We had two hypotheses. The first one is that since Foxo1 is known to repress cell proliferation in the blood endothelial cells, we will check whether proliferation plays a role in the new valve formation. Our second hypothesis is that as a transcription factor, Foxo1 could directly repress the expression of valve-forming genes such as FOXY2, GATA2, and PROX1. What I mean by that repressor is that if we remove Foxo1, one would expect that the expression of these valve-forming genes would be upregulated, and that would lead to new valve formation. So we tested these two hypotheses individually. We first check whether proliferation is involved in the new valve formation. We analyzed the mesentery by whole mount immunostaining with valve gene FOXY2 and the cell proliferation marker Ki67, and counted the number of Ki67 positive cells in the valves and in the lymphangium between the valves. As the graph shows here, we did not see a difference in the number of Ki67 positive cells between the Foxo1 lymphatic knockout and the controls. These data show that proliferation is not a part of the mechanisms that drives new valves to grow. Next, we investigated the transcriptional regulation of Foxo1 during valve formation. We knocked out Foxo1 in the cultured human lymphatic endothelial cells using shRNA and analyzed the expression levels of valve-forming genes using real-time PCR. We saw increased expression of many valve-forming genes, including FOXY2, GATA2, KL4, KF2, and KF2. And we saw this increased expression levels on both the mRNA level and the protein level in the Western blot results. Additionally, we treated these cells with oscillatory flow, and these genes were, I forgot to mention that also we treated these cells with oscillatory flow, and these genes were upregulated by oscillatory shear stress as previously reported. So when then we were wondering whether how Foxo1 activity was regulated by oscillatory shear stress. We treated these cells with oscillatory flow and stained Foxo1 to look at the cellular localization of Foxo1 protein using immunostaining. You can see that after 10 minutes of oscillatory flow treatment, FOXO1 lost its nuclear localization. And meanwhile, a Western blot showed the level of phosphorylated AKT and FOXO1 was upregulated under the same flow condition. Additionally, by treating these cells with an AKT activator SC79, we also saw an increase in the level of phosphorylated FOXO1. With this data, we were able to link shear stress, AKT, and FOXO1 in the lymphatic valve formation by showing that AKT is activated by shear stress, and AKT phosphorylates FOXO1 to decrease its nuclear activity. Since FOXO1 represses valve gene expression in the nucleus, when FOXO1 lost its nuclear activity, the expression of valve-forming genes was upregulated. Another important question was what the downstream target of FOXO1 is during lymphatic valve development. To answer this question, we performed chromatin immunoprecipitation assay to demonstrate that FOXO1 directly bound to the promoter region of the FOXC2 gene to transcriptionally regulate its expression. Although we only showed FOXC2 as a direct downstream target of FOXO1, we speculate that FOXO1 also directly regulates other valve-forming genes. To summarize these experiments, we have shown that FOXO1 represses valve formation by repressing the expression of valve-forming genes. Deletion of FOXO1 removed this repression activity and induced new valves to grow. In the rest of my talk, we explored the therapeutic value of FOXO1 deletion in a lymphedema animal model to answer the question, does FOXO1 deletion restore lymphatic valve defects in lymphedema? My mouse is a little bit jumpy, sorry. So to recap what I mentioned earlier, FOXC2 heterozygosity causes lymphedema distichiasis syndrome. Dermal backflow is seen in these patients, which indicates that lymphatic valves are defective. Therefore, we used FOXC2 heterozygous mice, which is the animal model for the human disease lymphedema distichiasis. This FOXC2 mouse model generated by Dr. Satish Srinivasan also has CRE-ERT2 activity. We crossed FOXC2 CRE-ERT2 mouse with FOXO1 Floflox mice, which allowed us to knock out FOXO1 in the FOXC2 heterozygous background. We analyzed the pups 3 weeks after deletion. We counted the number of valves, branches, and the valve number per millimeter in the control FOXC2 heterozygous with 1 allele deletion of FOXO1, and FOXC2 heterozygous with 2 alleles deletion of FOXO1. So as you can see here, FOXC2 heterozygous mice lost about 50% of valves compared to the control, although 1 allele deletion of FOXO1 did not rescue the valve number. Knocking out 2 alleles of FOXO1 restored the valve number and the branch number to control levels. To further confirm that the morphology of valves and the expression of valve genes were restored in the FOXC2 FOXO1 rescue animals, we performed the confocal imaging of PROX1 FOXC2, VE cadherin, and integrin for alpha 9. staining in these 4 groups of animals. We showed that although the defective valves in the FOXY2 heterozygous animal expressed PROX1 and FOXY2, they lost the valve leaflets shown by integrin for alpha-9 staining. However, the loss of valve leaflets and the expression of integrin for alpha-9 were restored by deleting FOXO1 in the FOXY2 heterozygous animals. Therefore, both the number and the morphology of defective lymphatic valves in FOXY2 heterozygous were restored to the control levels by deleting FOXO1. Then a more important question came up. Are these valves induced by FOXO1 deletion functioning as normal? To answer this question, we tested whether FOXO1 deletion can restore valve function in FOXY2 heterozygous mice by collaborating with Dr. Mike Davis at University of Missouri. As I mentioned earlier, healthy valves seal against the pressure to make sure lymph does not flow backwards. Dr. Davis developed this technique to test the ability of each valve to seal against pressure. And briefly, each lymphatic vessel was cannulated on 2 glass micropipettes. The output pressure was gradually raised while keeping the input pressure constant. A smaller pipette called a server-node pipette inserted in the vessel lumen right behind the valve leaflets measured the pressure leaking backwards through the valve leaflets. So if the valves are leaky, the server-node pipette measures the pressure to indicate how leaky the valves are. These are the representative picture of the valves from all 4 groups. When output pressure was elevated to a physiological pressure of 10 centimeters water, there was a noticeable pressure leak in the FOXY2 heterozygous animal. On the other hand, when either one allele deletion or 2 allele deletion of FOXO1 completely rescue this back leak in the FOXY2 heterozygous background, as you can see here. In addition, because subtle defects in valve sealing can be magnified at higher pressure, we tested the same valves to 100 centimeter water. At a pressure this high, some control valves eventually fail and begin to leak. However, none of the FOXO1 deletion valves showed any back leak even when the valves were facing a pressure of 100 centimeter water. These experiments showed that loss of FOXO1 significantly enhanced the valve function in a mouse model of lymphedema distichiasis. To summarize our findings, we identified the first transcription factor that represses lymphatic valve formation by repressing the expression of valve-forming genes. We showed that the oscillatory flow, AKT, FOXO1 signaling pathway is critical for lymphatic valve formation, and FOXO1 directly represses the expression of one of the key valve-forming genes, FOXY2. We were able to show deletion of FOXO1 in lymphatic endothelial cells can induce new valves to form and can rescue valve formation and function in the lymphedema mouse model, FOXY2 heterozygous mice. that has provided therapeutic implications for congenital lymphedema with valve defects. I would like to thank the people from my lab who worked hard together on this project. I would also like to thank my collaborators for their tremendous support. And I cannot complete this project without the support from my department, the imaging core facility, and the mouse facility. Lastly, I would like to thank NIH for funding support. Thank you everyone for coming to my talk. I will take questions from the Q&A box now. The first question is, so the FIT4 FLT4 VEGF receptor 3 mutation is not associated with valve defect then? I didn't see that gene on your list of affected gene associated with valve defects. So yes, this is absolutely right. So far we have not seen the evidence that VEGF receptor 3 regulates valve formation, but we cannot completely rule that out. But I know the scientists who are working on the VEGF receptor 3 mutations show that the lymphatic vasculature has other defects other than valve defects. For VEGF receptor 3 mutations, valve defect is not the major defect, I would say. So the next question is there a cure for lymphedema? So far there is not a cure for lymphedema, either for the congenital lymphedema or for secondary lymphedema. We actually understand more and more about both types of lymphedema. Although the exact cause for secondary lymphedema still remains unknown, but valve defect is a big component in both congenital lymphedema and the secondary lymphedema. And our work for FOXO1 inhibition could potentially develop into a cure for at least congenital lymphedema with valve defects. And the next question is, I am just a patient of secondary lymphedema for 25 years after my mastectomy, cannot understand this high-level info. Therefore, what is the practical suggestion for us to do after so many years of lymphedema? I don't think I can provide you very much of the treatment because I'm not a clinician. But my point would be keep good lymph flow will be beneficial for any lymphedema patients. And the next question is, I have primary bilateral lymphedema if in the legs and the trunk. How can one join this study? So far our study is still in the animal models, and we are trying to move into more pharmacological methods as the study progresses. And so far we have not actually moved into any human studies in the near future. But eventually we would like to develop some kind of treatment in the long run. The next question is compression the only treatment? So far, the compression would be the only treatment that any patients can practice at home. But there are other treatments like surgical treatment that you can consult with lymphedema expert physicians that they could answer the question better. Then the next question is, would a patient with 15 years of infections and scar tissue after 18 lymph nodes removal from breast cancer be a candidate for this potential treatment? So it's a kind of difficult question to answer because this is a situation for secondary lymphedema. And a long-term secondary lymphedema case. So because although valves are big components in also thermal backflow, which indicates valve defect, and is a big component in also secondary lymphedema, the deletion of Foxo3 or Foxo3 inhibition potentially would possibly work better in the earlier treatment. But enhancing valve function, because in my presentation, I also showed deletion of Foxo3 can actually enhance valve function as well, not only the morphology. So enhancing valve function will be another benefit that you can have from Foxo3 inhibition. So I can completely rule out the potential for this situation. The next question is there any research studies similar to this, but on human models? The short answer to this question is no. We are still working on the mouse models to actually better understanding how lymphatic valves form and how lymphatic valves function better. In either the Foxo3 knockout mouse models or in other mouse models. So next question is, what does this mean for long-term primary lymphedema patients? So this is a good question because the deletion of Foxo3 or Foxo3 inhibition, you could imagine would work better or more effective in the earlier, start from the earlier treatment. But since deletion of Foxo3 can also enhance the function of the lymphatic, of these lymphatic valves, and it, which means it has the potential to also benefit long-term primary lymphedema patients. The next question is, can anything be inferred from your congenital study to those with secondary lymphedema? This is also a very good question. Because for sure, valve defects caused by gene mutations are the causative factor for congenital lymphedema. So Foxo3 deletion can correct, can restore the valve defects in those mouse models, which is very encouraging. But meanwhile, what I mentioned in my talk was the secondary lymphedema patients also have dermal backflow, indicating valve defects. Our discovery could also potentially benefit the secondary lymphedema patients as well. And my laboratory actually is trying to understand more about the secondary lymphedema and see whether the secondary lymphedema mouse model could also benefit from the FOXO1 inhibition. The next question is, what potential do you think there is for a cure for primary lymphedema, particularly lymphedema distichiasis from FOXC2 gene in the next few decades? Would interventions of the FOXO1 ever work in individuals later in life? Example, not shortly after birth. So we actually deleted FOXO1 in the adult animals as well. And we found FOXO1 deletion also increased the valve formation in the adult animals. FOXO1 deletion also works after valves were actively forming. The difficult part, I would say, is that because FOXO1 is expressed in many other tissue types, so inhibiting or deletion of FOXO1 wouldn't be good for other tissue type. So cell-specific deletion or inhibition of FOXO1 for lymphatic endothelial cells would be something to work on if we want to move forward into the intervention of FOXO1 in human patients. So the next question is, are there drugs that can inhibit FOXO1? So far, there is no drugs that can inhibit FOXO1. In the lab, we have commercially available FOXO1 inhibitor that we are currently using that to study pharmacologically inhibiting FOXO1 in the animals, in the cells, to check the effect of this pharmacological inhibition of FOXO1. So the last question I would answer is, will primary lymphedema patients benefit from this research? How do you know if you have valve defects? Will treatment be available, and when would be the time frame? There are three small questions. Our study mainly showed deletion of FOXO1 actually make the primary lymphedema mouse model, the valves from the primary lymphedema mouse model function better. I would speculate the primary lymphedema patients with valve defects could potentially benefit from this study. And how do you know if you have valve defects? So for patients, their DNA sequencing that you can consult with experts that you need to could get information of any gene mutations you could potentially carry. And many genes have identified by DNA sequencing are related with primary lymphedema symptom. And many genes, we actually, my lab is not doing gene sequencing, but the experts have already identified more and more genes that are associated with primary lymphedema. And many more mouse models have made according to these mutations. So we have identified more and more genes that have valve defects that caused primary lymphedema in humans. So about the time frame, I don't have an answer to this question because any basic research actually takes a really a long time to analyze all the phenotype, evaluate the effect, and also side effects. So my laboratory particularly will be working hard every day to understand more of valves of Foxo1 deletion. And our long-term goal is eventually to develop some therapeutic treatment for lymphedema patients. I really hope what I do, what my laboratory is doing, would help lymphedema patients in the future. So thank you everyone for coming to my presentation. And I'm happy to share our results with you guys.